Excellency Tasha Sonam Topke, Ambassador of Bhutan to Australia, distinguished guests, esteemed speakers, and valued participants, a very good morning to you all. Welcome back to day 2 of the 3rd Australia-Bhutan Research Conference. It is wonderful to see you all here as we continue our journey of dialogue, discovery, and collaboration. For the new participants, my name is Tenzin Chodin, and together with my colleague, Madam Siring Chodin, we will guide you through today's session. I'll be your MC for the morning session of the program, and it's my pleasure to welcome you. We begin this morning with Thematic Area 3, Integration of STEM for Societal Impact. This thematic area highlights how interdisciplinary STEM research is addressing real-world challenges, from biosecurity and genomics to sustainable construction materials, biodiversity management, and cutting-edge microfluidics technologies. It demonstrates how scientific innovation can directly enhance resilience, improve health systems, and contribute to sustainable development in Bhutan and beyond. A reminder to our presenters, you have a total of 12 minutes for your presentation. You will hear a bell when there is 2 minutes left of the presentation time, a signal to wrap up, and a final bell again to indicate that the presentation time is over. Our first session opens with a keynote address by Professor Kentai Yong, Associate Dean for External Engagement at the Faculty of Engineering, the University of Sydney. Professor Yong is a professor of biomedical engineering and a senior member of the National Academy of Inventors. His multidisciplinary work spans microfluidics, nanomedicine, biosensing, and quantum dot engineering, translating fundamental science into healthcare innovation. With over 300 publications and more than 25,000 citations, he has received the Bealby and the Rosenhain medals. Professor Yong is also a co-founder of the Net Zero Initiative, leading research in climate innovation and governance, and is a fellow of several prestigious scientific academies. This morning, he speaks on the topic Microfluidics and Organ-on-Chip Technologies, Reimagining Innovation, Health, and Well-being for a Sustainable Future. Please join me in welcoming Professor Yong.
Good morning, everyone. Kuzu Zongpala, Vice Chancellor of RUB, His Excellency Sonam Topge, distinguished guests, colleagues, and friends. It is actually an honor to join you here in Bhutan, a place where science, compassions. Yep. It's an honor to join you here in Bhutan, a place where science and compassion's human dignity naturally come together. So every time when I actually visit Bhutan, I'm reminded of how innovations must ultimately serve people, especially those living in the remote mountains, valleys, scattered communities that make Bhutan so unique, resilient, and beautiful. So today, I want to share a vision how microfluidics and organ-on-a-chip technologies can help reimagine healthcare, well-being, not just scientific milestones, but as a tool for sustainability and hope. So let me brief you a little bit about the challenge we face today. So around the world, healthcare is actually becoming more specialized, more centralized, and more expensive. But in countries with challenging geography like Bhutan, distance can become a life and death barrier. So a simple and diagnostic test may require hours of travels. Samples degrade on the journey to a central lab. Remote clinics lack sophisticated equipments. Veterinary cares for the rural livelihoods face similar bottlenecks as well. So these challenges are common across mountain nations, but they inspire an important questions. So what if the lab could be miniaturized into a palm of your hand? Or what if an entire organ systems could be replicated on a small chip size of a credit card? So this is actually where the microfluidic and organ on a chips technologies enter the story for today's. Now, microfluidics and organ on chip. So what are they really? So microfluidics is actually a science of manipulating tiny volumes of fluids, microliters or even nanoliters inside the channels tiny than a human hair. So from these foundations, it's kind of spin off to two very transformative technologies. The first one is actually lab on a chip. We call it LOCs, which is a portable, rapid, efficient versions of a lab test. And it allows diagnostic at the point of care, small sample volumes, fast turnaround time. And the second one is actually the organ on the chips. We call it OOC. And this is actually where engineering meets biology itself. So OOC devices, they're actually mimicking the physicals and biochemicals environment of human organs. The lung, liver, gut, heart, skin, many more. Currently, if you Google a little bit, there'll be many of those, the organ on the chips. So these chips maintain the living cells, continuous nutrient flow, mechanical forces, and real physiological interactions. And more so, they allow us to test drug ethically without animals, study disease much more realistically, and more important, so personalized treatment itself. So currently, as we speak, organ on a chip systems are now supported by the America FDA Modernizations Act 2.0 and 3.0, which encourages non-animal testing for drug developments. So this is actually a big plus right now. So this is actually a profound shift, one that Bhutan can align with early. Now let's discuss a bit about my research journey. So for the past decade, my groups have been working to push the boundaries of microfluidics, lab on a chips, and organ on a chips technologies. So allow me to share a few examples, not in a complex scientific language, but in terms of their impact. So I'm going to briefly just go through some of this and share with you some of this, the images and our experiments. The first one is actually cell sorting with initials microfluidics. So we engineer microchannel that use fluid forces to automatically sort the cells. So this is actually, think about it, the flow cytometers that's actually used in clinics. Lab on a chip systems for drug screenings, nanoparticle synthesis. We create platforms that synthesize nanoparticles and test therapeutics within the tiny fluidic devices. For example, liposomes, nanoparticles for anti-cancer drugs therapies itself. So this means that we are actually able to produce safer drugs evaluations, reduce costs, higher precisions. And the third one is actually MEMS. Basically, it's actually mechanical electrodes, miniaturized systems, implantable drug delivery device, where I'm actually going to share with you some examples as well. So this is actually one of our most meaningful projects involved building on a miniaturized implant that release drugs within high precisions, powered only by micro actuators or even by biokinetic energies, from the motions of the patient's movements. So these technologies could one day help patients with chronic disease who need low consistent medications. We also work on lung on a chip for toxicologies. We replicate human lung environments on the chip and to study how cigarette smokes cause cells transformations. This research demonstrates that how organ on the chips can review disease in the mechanism way with clarity that animal models cannot match. And more recently, our latest work, liver on the chips and animal on the chips models, we are now building a biomimetic liver lobule and a drug testing system for the dog bone cancers therapies, and of course, with multi-cell interactions models involving cancer cells and macrophage. So this is not just science, it is actually a beginning of a personalized medicines for both human and animals itself. So imagine a world where a dog with the bone cancers in Bhutan could have its cells tested on a chip, and so that the vet colleagues knows exactly which treatments will work the best. So this is actually not distance. This is actually happening now. And with that, and let me share with you some of these examples. So this is the miniaturized systems over here. As you can see, it's actually the microfluidic channel itself is like in the tip of your finger. So this is actually the lab on a chip for flow cytometers, and flow cytometers are generally used for counting the red blood cells, white blood cells, and many of those. Again, currently we are able to make it miniaturize it within a very small chip itself, and by using a simple setups, quite complicated, but we are actually able to do it using a much smaller platform right now by sorting all this, counting all these various kinds of particle cells and disease tumors cells as well. And we are actually able to implant, like I've actually mentioned, the devices into using the biokinetic energies of the small mice, and they're actually able to deliver the drugs. So we have actually tested it, and it's actually biocompatible. More importantly, right now, they are able to provide a good drug efficacies, onto the tumor-bearing mice itself. And this is actually a standalone lab-on-a-chip systems, basically helping us to evaluate multiple drugs, because to treat one disease itself, you need more than one drugs to apply on it itself. So this system, we're actually able to automate it. So basically, we centered the tumor cells in the cell culture chamber in the figure D, and then subsequently, we're actually able to pump in formulation A, B, C, D, and it will actually continues to test it until it finds the correct dosage. And from there onwards, we can actually engage our clinicians and inform them saying that this A with this dosage, B with this dosage, C with this dosage, plus together, it has the maximum highest impact on this particular cancer cells. And we are also developing, working very closely with our colleagues on this a new nano battery. There's actually a term called triboelectric nano generators. So basically, nano batteries where it can use the hand movement to power enough of electricity to pump the FDA-approved drugs to treat glaucoma. So we are actually currently, finished the prototype, and right now we are actually working together with our hospitals colleagues, to move on to the next animal phase. So this is actually the liver on a chip that we actually mentioned. We are actually designing this is a work in progress, but I think that's actually very interesting. Why? Because liver is actually one of the system to cleanse our body from the toxins and various kinds of chemical itself. So currently, we are actually trying to place all these liver tissues in the chip, as you can see, where all these various kinds of nutrients we are actually trying to mimic from the body parts, and able to making sure that they are having a very similar physiological impact and functions, where it actually allowing us to understand their impact. So maybe I can skip all this. If you're interested, we can discuss more. But let me continue a little bit, about why this matter for Bhutan's. So let us now connect these technologies to Bhutan national visions. So the first thing I want to discuss about the healthcare in the mountains, because microfluidic devices, they are small, portable, low power, low cost, and require only tiny sample volumes. So this aligns perfectly with Bhutan needs. Remote clinics with limited equipments, difficult terrains, long travel distance, limited cold change capacities. So a lab on the chips device could allow the health worker in Laya to test liver functions, infectious markers, or even cancer biomarkers within minutes. And the second one is actually the veterinary and livestock health. So the rural livelihoods depends on heavily on animals. So the animal on the chips models and multi-cells microfluidic platforms can inspire the livestock disease testings and drug sensitive of profilings and rapid screenings systems for veterinary use. And the sustainable and ethical innovations often on the chip systems can reduce animal testings and aligning strongly with Bhutanese values of compassion and well-being. And building Bhutan's for future STEM workforce, this platform create unique opportunities for training opportunities, hand-on engineering skills, biotechnologies exposures, research pathway for RUB students. Bhutan is actually growing its digital STEM and innovations ecosystems. So microfluidics and OOC can be one of the pillars of that ecosystem itself. And I would also like to touch upon on the Bhutan as a global test beds. Because of its geography and skills, Bhutan can leapfrog traditional systems and become a model nations for frugal innovations for remote healthcare. So this can attract a couple of things. One, global collaborators. Two, grants, the entrepreneurship, startups, and new R&D partnership as well. And let me discuss a little bit the way forward. So for me, I think that the innovation is actually not just about technologies alone. I think it's actually about people. So together, I think that we can build a Bhutan Australia microfluidics or Sydney microfluidics overseas innovations corridor through joint labs, student exchange, capacity buildings, co-developed devices for remote clinics, and collaborative high impact papers as well. And of course, about the pilot projects and remote diagnostics, a few microfluidic-based prototypes can already be tested in the high altitude areas, basic health units, mobile clinics, veterinary facilities, and more so about a national roadmap for ethical drug testing as well. So with global the regulatory shift, Bhutan can adopt a non-animal testing technologies early. And not to mention about talent developments, short courses, workshops, and research internships can empower a new generations of Bhutanese engineers and scientists itself. So in closing, let me end with this thought itself. So when science becomes small enough to fit onto a chip, its impact becomes enormous. So it can reach a yak in the mountain valleys, a child in a remote village, a monk in the distant monasteries, or a loved one who needs a diagnostic now but not tomorrow. So microfluidics and organ on the chips technologies represent not just the futures of biomedical engineering, they represent a futures where healthcare is more humane, more accessible, and more just. So Bhutan has all the ingredients that I believe to lead to the next wave of innovations. And I'm deeply committed to walking these journeys with all my Bhutanese colleagues, scientifically, collaboratively, and wholeheartedly. Thank you. Tashi Tale.
Thank you, Professor, for your profound reminder that innovations must serve people and the opportunities that you highlighted for Bhutan in this field. Our next presenter is Tawang Nidup, a faculty member of College of Science and Technology, specializing in structural design and civil engineering project management. He has contributed to national engineering standards and quality control with a strong focus on sustainable nature-based solutions. His current research explores timber engineering and structural reliability, including timber strength grading for structural applications. His presentation is titled Attributes of Bhutanese Conifer Timber for Optimal and Sustainable Application in Building and Construction Sector.
Your Excellency, Dr. Kusap, distinguished guests and participants from near and far, a very good morning. Let me begin by sharing some images to set the tone for my presentation today. So my presentation today is a story about what we need to know and how we go about knowing what we need to know about our timber resource to make use of them, to convert them into a building that we have just seen. So the timber and the building industry presents a very unique and mutually beneficial opportunity. Timber presents a very viable means to decarbonize the building sector. And being a renewable and natural material with proper forest management practice in place, it is in principle possible to have an unlimited supply of timber stock. The building sector, on the other hand, presents the best avenue and scope for high value utilization of this resource. So given these benefits, timber is increasingly being held as the building material of the future. Now, our country is one of the few countries in the world with more than 70% of its area under forest coverage. So in fact, we have one of the highest per capita timber resource in the world. So given this abundance, Bhutan, I think, is well placed to harness the technical, economical, and environmental benefit of using our timber resource in the building sector. However, the resource currently is severely under and misutilized. The annual increment of new wood stock in our forest is way much lower compared to some regions and countries around the world. Furthermore, the annual harvest rate is a minor fraction of this already very low increment rate. A classic example of the misutilization of this resource is the use of wood as a material to make formwork in concreting, rendering premium quality timber completely and functionally useless in the process thereafter. Now, one of the reasons hindering the use of timber is the complete lack of knowledge and data about its character critical for its use as a building material, such as its strength, its durability, its performance in fire, and its the ease with which we can treat and season timber. The timber from a forest have never been scientifically investigated for such purpose. And therefore, the parameters pertaining to these properties at the moment are anybody's guess. Now, in effect, timber, like any other building material, can be properly characterized. And this is what's happening elsewhere in the world. The label and the image that you see here is an example for Norway spruce in Europe. So this label 24, C24, contains and provides information related to key strength and other physical and mechanical properties of the species. Likewise, the information pertaining to its performance and reaction in fire, its durability class, and the particular timber species amenability to seasoning and treatment. Based on certain basic mechanical and physical properties, timber can be categorized into appropriate strength class. So the example that we see here, is for spruce growing in the UK, has been found to meet the requirement of C24 strength class. Now in practice, timber strength grading can be done either visually or through machines. In the visual grading process, certain anatomical feature of the timber piece, like knots, the rate of growth, and the slope of grain on a piece of timber can be used to assign visual strength grade class. So for example, a large species growing in the UK graded according to the UK visual grading rule has been assigned a visual class SS, which has been found to meet the requirement of the strength class C24. Likewise, now there are machines available to grade to strength the timber, resulting in a much more improved efficiency and accuracy of the grading process. However, again, both the rules for the visual grading process and the settings for the machines needs to be based on and validated through extensive standardized testing of sufficient number of representative timber samples. So today, what I present here are the key findings on 2 conifer species growing in the Gidakum region in Thimphu, tested as a part of the overall investigation campaign to develop a national timber database in the country. The 2 species I'm presenting here are for fir and hemlock. The investigation itself are based on established international standards and norms. And I share this image. I think it's a very special image because probably I think this is the first ever piece of native timber species being tested for such purposes. So the date was 19th March 2025. So now coming to the findings and results, to start off with the anatomical feature, On the north, the maximum knot area was about 50% of the cross section from amongst the tested pool of specimen. And this would qualify the visual grade S7K of the German visual grading rule. Coming to the rate of growth, the maximum width of growth ring was found to be 5 mm, which would qualify the visual grade S10K of the German visual grading rule. Coming to the mechanical properties, the characteristic density was found to be about 362 kgs per meter cube, the bending strength about 14 megapascal. And comparing that to the example that we have seen earlier on British spruce, while the density value more than met the requirement for C24, in this particular case, the limiting parameter was the bending strength, which limited the strength assignment to C14. In the investigation, the samples were taken from different positions along the tree height. So some of the specimens were from the bottom, some were from the middle, some were from the top. And the findings indicate that the specimens from the bottom are usually more stronger than ones taken from the top. And probably the reason for this is, if you look at this tree architecture, as we go up higher along the tree, there are more branches. And knots are one of the growth feature that is quite detrimental to timber strength. Now, talking about the basis for the formulation of a visual grading rule for native timber species, as you can see here on the image, the strength is inversely related to the size of the knot. Likewise, it's also inversely related to the width of the growth ring. So based on the correlation that we can develop between this anatomical feature and the strength of the timber, a basis for the formulation of species-specific grading rule can be established. Likewise, for the machine grading, the stiffness of the timber, so in layman's term, it kind of indicates how flexible or how inflexible a material is. So it has been found to be the best indicator of timber strength. And the good thing about that is there are now machines that can be used to measure and determine the stiffness. So the work established the settings that will be required for these machines, which later we can use in our sawmills or in other timber manufacturing facility, is also underway. So yes, in conclusion, we all know that the rivers in our country are fondly regarded as the white gold. So with the right ecosystem in place, I think it's not too far-fetched to say that there's every possibility for the forest and the timber resource to be the green gold. Finally, I would like to conclude by this quote, maybe a bit controversial and also a bit counterintuitive at first glance, that the use of timber in construction, perhaps the only chance to save the forests of the world. Lester, thank you, and Tashtile.
Yes, thank you, Mr. Sawang, for a very interesting presentation highlighting timber as the building material for the 21st century and how Bhutan can tap into this potential, as well as for kickstarting this with your research lab. Now, our next presenter is Dr. Mamta Chhetri from the University of Sydney. Dr. Mamta is a serial pathologist and a molecular geneticist at the University of Sydney's Plant Breeding Institute. She leads Australia's National Serial Rust Surveillance Project and manages quarantine diagnostics for imported germplasm. Her research focuses on pathogen genomics, rust-resistant genetics, and disease surveillance. She will present on integrated surveillance and biosecurity strategies for plant pathogens, lessons from Australia for Bhutan. Please welcome Dr. Mamta. Honorable, Vice Chancellor, and distinguished colleague,
I'll be presenting on integrated surveillance and biosecurity strategy for plant pathogen, lesson from Australia to Bhutan. So if I go into detail about what plant pathogen are one of the biggest challenge to global food security. Among them, rust disease is the devastating and destructive disease posing a serious threat to food security and biosecurity. There are about 3 cereal rust. You can see from here, stripe rust is yellow in color, and leaf rust you can see by brown pustules, and stem rust is a black pustule. And this can infect cereals like wheat, barley, and oats. At our institute, we also have a collection of spores dating back to 1950s, which we use it for our research purposes, stored in liquid nitrogen. And rust spores, when it comes on the leaf surface, it lands on the host leaf, then they enter the stomata, causes infection, and form pustule that releases billions of spores spreading rapidly on a wind current. So if I give you a background of rust, I'm going to talk about COVID-19. Everyone must be knowing about COVID-19, so I'll relate to that. And rust spread, much like COVID-19, through wind and human activities. This image you can see from there, there is a tractor there, and then rust spores are spreading in the field. Just like COVID-19, when you sneeze, the droplets transmits the disease. It's similar. Under high infection pressure, mutation occurs, creating new variants or pathotype that overcomes plant-resistant genes in rust. So from this picture, you can see that side, it is rusted seeds, and there is a healthy seed. So you can see that this is also similar to the COVID-19. So, and this is the picture during rust epidemic in Australia, where it caused about 80 to 90% yield loss and over $100 million in annual damage in wheat alone, which came out in a local newspaper. So, continued surveillance and early warning system are critical to prevent these outbreaks. So that is what we do at the University of Sydney. And just like in COVID-19, exotic incursion happened, and they moved through air, hitchhiking on people. Even the microscopic amount of rust, you can see from this picture, people have traveled to the rusted areas and they're carrying spores in their feet. So rust spores not only travel through human clothing, but also can travel thousands of kilometers across continent on the wind. This you can see that rust spore is spread through wind dispersion. In Australia, we have had a major exotic incursion, barley leaf rust and wheat stem rust, and we had about 4 stripe rust incursion from Europe, America, and other areas. These causes was that, the rust doesn't respect boundaries. That's why even a microscopic amount can trigger an exotic incursion. Once it is in, it spreads fast, just like COVID-19 did. That is why strict biosecurity and rapid response is very essential. So at the University of Sydney, Australian Cereal Rust Control Program based at the University of Sydney and funded by Grain Research and Development Corporation since 1921. It's been a century. It's one of the world's oldest and most coordinated biosecurity program. It's been protecting crops over centuries and works closely with 6 partner institutes. We are now expanding this collaboration even further. So the basic focus on 4 key objectives we have. First one is strengthening the national surveillance using field monitoring, molecular diagnostic, and genomic tools. The second one is enhancing genetic resistance. Why do we require this? Because there are new variants coming in and we have to resist the new variants. So identifying and deploying durable resistant gene is vital for breeding programs. And the next one is boosting biosecurity preparedness through Department of Agriculture and Fishery and Forestry, Quarantine Compliance, and Rapid Response System. And next one is at the University of Sydney, we train next generation of scientists, connecting researcher and institute across Australia. So if I go into the detail about the national cereal rust surveillance in Australia, this pillars keeps Australia ahead of pathogen evolution and serves as a model. So early detection is the foundation of disease control and crop protection. Australia's National Cereal Rust Surveillance Program, led by the University of Sydney Plant Breeding Institute, receives samples from growers, agronomists, and researchers across all major cereal growing areas, just like COVID samples. And throughout the season, cereal rust lab at the university analyzes stripe rust, leaf rust, stem rust, oat samples for pathotype identification. On this map, you can see yellow stripe rust, and while other colors are different rust. We produce this map continuously throughout the season so the growers can track pathogen spread in real time. It's a critical tool for making timely decision on fungicide use and also varietal selection. So what methodology do we use? So rust pathogen mutate rapidly, producing new pathotype that overcomes resistant genes, just like in COVID-19 evolves. On the left, you can see 3 different pathotypes infecting wheat variety with different resistant genes inside. Each reacts differently. The second pathotype have overcome some of the genes in that wheat variety, and third pathotype is really virulent and it has overcome most of the genes. So on the right, we also do molecular tool to identify rust at DNA level. This allows us to classify them at a lineage level. And however, within a lineage, it is much harder to distinguish different variants within the lineage. That's why we use traditional pathotyping and molecular analysis to accurately identify which variant is going on in the field. So managing this is critical because high infection pressure accelerate mutation and there will be new variants developing. So genetic control and reducing disease level saves Australian $1 billion of wheat and cereal industries. So if I go into the detail of results, every year we receive 1,700 samples or more each year. It is all dependent on the climatic condition and conduciveness of the rust disease. So in 2024, 198 pathotype was dominant, declining, just like omnivariant declined when Delta variant came in. And then after that, what happened is like 299 variant came in, which dominated, and Delta variant it's just like Delta variant kind of thing. We also identified new variants, 238 variant, which was found in Queensland and New South Wales. Another variant we identified was BGYR lineage, which was identified in 1998, but it was only infecting barley grass. Now we have identified two new variants which can infect barley and wheat. And I have communicated this issue to grain growing community. So monitoring this variant is very critical to guide Australian grain industry for breeding and fungicide management. We also do fungicide resistant treatment. This in fungicide resistant, from this graph we have identified barley leaf rust variant which can resist fungicide treatment. So that is, sorry. That is the one, like, I mean, even at 25x of fungicide treatment, it is still, like, I mean, alive, the fungus. So I will just go into the key message before I'll just leave this part away. And this fungicide resistant isolate is all over Australia. So I will, and we also did genomic analysis, and what we found was sensitive, like, I mean, which can isolate, which can be killed by fungicide treatment, and insensitive isolate. There was in the gene CYP51 locus in chromosome 9, there was a copy number variation which caused the resistance to fungicide. Last, my only for preparedness plan for Bhutan, I would like to recommend strengthening Quarantine Act, quarantine facility, and integration of lab agriculture ministry so that it can create a linkage just like in Australia. That's my recommendation for Bhutan. Thank you.
Yes. Thank you, Dr. Mamta, for your presentation where we learned about plant pathogens as an increasing biosecurity threat and recommendations for Bhutanla. Our next presentation comes from Dr. Leela Reisel, who will be joining us online. Dr. Leela is a mycologist whose work spans microbial ecology, biotechnology, and STEM education, guided by Bhutanese values of resilience and cultural rootedness. Her research journey began in Thailand, studying fungi under a royal wedding scholarship, continued with a PhD at the University of Queensland exploring insect fungi, and expanded into medical mycology at the University of Exeter, UK. Currently at CSIRO in Australia, she develops synthetic microbial communities to enhance soil health and suppress pathogens. Her topic today is genomic analysis and mating tests among sympatric isolates of Bulgaria bassiana provide evidence for a diverse cryptic species complex. Dr. Leela, can you hear me?
Hello?
Hello, doctor, can you hear me?
Yes, La. Can you hear me?
Yes, La, doctor.
Thank you.
You can start the presentation, La. Thank you.
Thank you very much. Good morning, everyone. I'm Leela Rizal, a mycologist at CSIRO, Canberra. Thank you for this wonderful opportunity. Today, I'll be sharing my PhD research from the University of Queensland on the genomic and mating biology of Bulgaria bassiana, a global important insect pathogenic fungus. This work is highly relevant because it uncovers a unique evolutionary link between the Himalayan insect fungus Ophiocordyceps sinensis, we called it Yaksa gembuk, and Australian Bivaria bassiana, two organisms with remarkable ecology and biocontrol significance. In the introduction, I would like to share some common things that we know about this entomopathogenic fungi. Entomopathogenic fungi are those that infect insects, but they do so differently. Some are specialists infecting only specific insects, like Ophiocordyceps sinensis, which target ghost moth, and they are very expensive. Others are generalists, such as Bivaria bassiana, and Metarhizium species, which can infect a wide range of insect species. Entomopathogenic fungi are either sexual or asexual. Asexual type penetrate the insect cuticle, break down chitin, and spread via spores, making them powerful insect biocontrol agents. Sexual type do the same, but also form fruiting bodies with sexual spores like Ophiocordyceps sinensis, prized in Asia as traditional medicine. Coming to the culture and economic significance, as you know, Bhutanese communities harvest Ophiocordyceps sinensis, one of the world's most valuable natural resources. To protect this prized fungus and its fragile alpine habitat, Bhutan requests permit even for locals. Harvesting is no easy task. It grows hidden among grass on the ghost moth larvae in harsh highland where collectors risk their life for every piece. So why is Ophiocordyceps sinensis so sought after? Because it grows only in a few Himalayan regions, and in fact, just one species of insect, the ghost moth. In Asia, it's prized as a traditional medicine, while in the West, it is hyped as Himalayan Viagra. And some even claim it the secret to eternal beauty. But fame has a price. Over-harvesting is exploiting it to the brink. Here you can see Bhutanese collectors selling their harvest at an auction yard. On the other side, a Facebook post claims to sell Ophiocordyceps sinensis, but it is not even the same fungus. Today, all kinds of products are marketed under the name Ophiocordyceps sinensis, highlighting the growing challenge of authenticity and mislabeling. Science has identified several bioactive compounds in Ophiocordyceps sinensis with anti-cancer, antioxidant, immunomodulating, and other pharmacological activities. However, there is currently no record of Ophiocordyceps sinensis being authorized as a novel food for general food use by the European Food Safety Authority. Now moving to Bivaria bassiana, a close relative of cordyceps found in Australia and worldwide. It's an entomopathogenic fungus widely used as an insect biocontrol agent. During my time in Queensland Department of Agriculture, I worked with Stephen Rice, another passionate mate of this fungus, to produce Bivaria bassiana insecticide, now being trialed to control chicken mites, especially laser mealworm in poultry farms. The laser mealworm is a notorious pest that damages poultry facilities, causing economic loss, and it spread pathogen to chickens and humans. Chemical control is failing due to resistance, but the fungus Bivaria bassiana offers an effective biological solution. We have successfully produced Bivaria bassiana-based biocontrol product in the lab, sufficient for farm trials. Below are the procedure used to generate the biocontrol agent. This photo illustrates the application of Bivaria bassiana-based biocontrol product in poultry sets, where the fungal suspension is being sprayed onto surfaces of chicken shed to target laser mealworm infestations. Field trials showed that Bivaria bassiana, highlighted by yellow line, achieved higher laser mealworm mortality than chemical insecticides. Although these fungi have enormous potential, there is a major research gap in understanding their identity, diversity, and sexuality, traits critical for biocontrol success. Traditional molecular methods using short gene fragment lack resolution. To address this, my research used whole genome sequencing and mating assays to uncover hidden diversity and sexual potential in Biberia bassiana. Recognizing the potential of Biberia bassiana as insect biocontrol and inspired by the value of Yaksa Gembo back home in Bhutan, my proposal received full Australian government scholarship in 2018 to pursue PhD at the University of Queensland, focusing on the diversity and virulence of subtropical entomopathogenic fungi. Moving to the methodology, unlike previous studies that sampled agricultural soil, I collected entomopathogenic fungi from the backyard, from the urban garden soil. This photo shows me sampling soil in an urban garden in a town called Toowong in Brisbane. I use insect bait such as diamondback moth, flower beetle, and cotton strinner bug on soil samples to isolate diverse entomopathogenic fungi, including Biberia and Metarhizium. Both are the most widely used bioinsecticide agent globally. I then assessed their virulence against diamondback moth larvae, a major pest of crucifer crops in Australia, and found them highly effective for control. These results were published. Knowing their potential as powerful insect biocontrol agent, I investigated the diversity of entomopathogenic fungi using whole genome sequencing. This photo shows part of that work in the molecular lab at the University of Queensland. Previous multilocus phylogenes group Metarhizium robertsii isolate together, as you can see on the left-hand side of this slide. But whole genome sequencing at the right-hand side revealed distinct genetic cluster, indicating hidden cryptic diversity. Similarly, whole genome analysis of Biberia bassiana, a widely used biocontrol fungus, revealed hidden genetic diversity, challenging previous assumptions. The finding on hidden diversity in the two most widely used entomopathogenic fungal biocontrol were published. During the COVID-19 lab shutdown, I accidentally left Biberia bassiana culture in an incubator. 3 months later, I returned to find floating bodies, a rare event for this asexual fungus. That unexpected discovery opened the door to deeper research into its sexuality and genetic diversity. I used mating type genes and whole genome sequencing and found that Biberia bassiana, though classified under a single name, actually form distinct genetic clusters, with isolate in each cluster significantly different from one another. Further, I used nuclear and mitochondrial genome phylogeny to compare global Biberia bassiana diversity and found Queensland isolate form a distinct clade, significantly different from population in other countries. Knowing that they were different from other global populations, I cross-isolate from the same and the different genetic cluster using mating genes and found that those within the same genetic cluster produce floating bodies on different culturing media and brown rice, similar to their relative Ophiocordyceps and Cordyceps. Biberia bassiana, the most widely used insect biocontrol, contain 4 cryptic species in Queensland, distinct from global population. Sexual recombination is extremely rare and occur only within the same genetic cluster. This finding raised an important question about its sexuality and suitability as a biocontrol agent in Australia. Developing optimized crossing protocols and floating body induction for Biberia bassiana would open 2 big opportunities. First, producing high-value fungal metabolites for Australia's pharmacological industry. And second, applying advanced culturing, ecology, and genomic techniques to boost natural productivity of Bhutan's prized Ophiocordyceps sinensis. Reference for these slides come from 3 leading fungal papers that were published. Currently, I'm working as a scientist at CSIRO. I'm developing a synthetic microbial communities to manage stubble-borne disease in grain crop. In Australia, Bhutan's agriculture relies on organic farming and natural biocontrol agents, and I'm happy to share insight on developing fungal biocontrol suited to our climate and discussing insight on the ecology of Ophiocordyceps sinensis. I'm open to collaboration on bioinsecticide and research. Please reach out via email. Thank you for listening, and I am very happy to take questions. And lastly, not the least, thank you Bhutan Embassy and Royal University of Bhutan for accommodating me for the online presentation. Thank you very much.
Yes. Thank you, Dr. Leela, for your presentations on our cordyceps and on Beauveria bassiana as a biocontrol agent. Please remain online as we will see you again shortly during our Q&A session now. Our last presenter for this session and thematic area is Samten Hendup, a faculty member in the Department of Mechanical Engineering at Jiminamgal Engineering College, where he has been teaching since 2004. He has made significant contributions to teaching, research, and academic development. His research focuses on building energy, sustainable design, and energy efficiency, exploring strategies to reduce energy consumption in buildings. In addition, he's actively involved in externally funded projects and student exchange programs with European universities. He will present on comparative study on thermal performance of mud rammed and concrete residential building envelopes in Paro, Bhutan.
Honorable Dashu Kutsak, distinguished guests, ladies and gentlemen, and all the viewers, online viewers, good morning and welcome to this presentation. This presentation is on the research-based project carried out by 3 students. These students are Mr. Ugen Wangchuk, Mr. Choten Singh Tamang, and Mr. Pemarinzin. Mr. Choten and Ugen are currently pursuing studies in sustainable and renewable energy in Australia, whereas Mr. Pemarinzin is currently serving as the assistant engineer in Mangdichu hydropower project. Actually, I have requested Mr. Pema to attend the conference and also do the presentation as well. However, due to his tight schedule, he could not make it. Therefore, I am presenting their work on their behalf. And to introduce myself, I'm Samten Hendup, working as faculty in Jigminamgal Engineering College under Mechanical Engineering Department. So their topic is on the comparative study on thermal performance of mud rammed and concrete residential building in Paro. The selection of their study site in Paro is primarily because of the availability of weather data from the Paro International Airport. So before proceeding, I would like to make the brief disclaimer. This research-based project focuses exclusively on energy performance, and it does not cover any aspects of the structural design. Now to begin, so the thermal performance of the building envelope is the key important aspects in determining the building energy efficiency. In Bhutan, the building energy sector accounts 15% of the total domestic energy electricity consumption. Again, 50 to 60% accounts for used for heating the space in the temperate region. So this this study also actually to evaluate the thermal performance of mud, ramped wall, and then concrete building, with the aim to enhance energy efficiency and sustainability through building envelope optimization. Now, on the background, as we all know, traditional Buddhist architecture uses locally sourced mud wall, offering good insulation as well as good thermal performance. Now, but what we can understand is modern concrete structures are becoming more common, potentially increasing the energy consumption for heating and cooling. On the research objectives, so there are three objectives: compare performance, energy saving, sustainable design. So by using the energy performance tools, they were able to evaluate the thermal performance between the mud ramp and then concrete walls, and also able to compare their performance in terms of values. And then as an outcome of the simulation, so it's able to quantify the potential energy savings achieving using the traditional mud rammed walls in constructions. And then at the last, it's able to promote the benefits of using local materials, enhance sustainability in the building practices in Bhutan. So significance of the study, sustainability, so supports Bhutan's goal for sustainable development and energy independence, ensuring a greener future. On the energy efficiency, reduces heating load significantly, promoting cost savings and decreased energy consumption in the residential building. Then third one, on the local materials, promote the use of traditional materials, enhancing local economics and cultural heritage in construction practices. On the literature review, many literature shows that 40 to 60% the energy losses depend on the building envelope. However, there remains a limited understanding how building envelope performs in the cold and high altitude climates such as Bhutan. Therefore, this highlights the need for localized assessment of envelope performance under the different climatic and operational conditions. With regards to the key theoretical framework, so the software that they have used was the SketchUp with OpenStudio. The reason is very simple, since that during that time the software was free. Now even SketchUp is not free, we need to buy. So the SketchUp is basically used as the graphics user interface for developing the civil drawings in the SketchUp. And then OpenStudio provides the provision where we can provide the inputs of the building details like construction type, material type, and then the how many people are living in that, and then also the lighting or the internal heat sources that they are available in that building. So all those the parameters can be provided through the OpenStudio. At the back end, Energy Plus provides the detailed numerical modeling, assessing energy demands and saving across different wall materials. So then other one, the important is that we need to have the weather data. So the whatever the weather data that we have used is obtained from the Paro International Airport weather station. With regards to the research gaps identified, there are three: comparative data, then empirical validation, knowledge shortage. Now, on the comparative data, there's a lack of comparative data between the thermal performance of mud rammed and concrete structures in Bhutan. Then the next, the empirical validation. The limited empirical validation exists for the building materials, indicating the need for more accurate, localized studies to substantiate the claim. Then on the knowledge shortage, the current research does not adequately address the knowledge shortage regarding the long-term performance of the traditional building materials. Now, methodology. So they have used the building simulation tools. So the input data that they have provided are the materials data, climatic data, and then design data, building usage data. And then output is the thermal load that's coming on the building. Then these are just information of the climatic data of the place. The exact location of their study site is in Paro Bongde. So these are the information that they have used as the input data. Now on the material usage, let's forget about the urban area, since we all know, or what we see around is that all the urban areas are converted into concrete jungles. Whereas even now the other worrying thing is that even in the rural area, now they started like converting into the concrete jungles. So from the data, what you can see is in the year 2007, the concrete usage was 9.5%, whereas in the 2012, it has increased to 17.3. It's almost increased by 8%. So now we can also understand from here that even in rural areas now started converting into concrete jungles. On the findings, the heating load dominates over the cooling. From this graph, what we can understand is that in the month of June, July, August, maybe they request a little bit of cooling energy. Otherwise, the other months are all what is required is for the heating. And then through our experience also, we know that the mud rammed buildings usually feel warmer than the concrete buildings. Just to support that, I think experience, even the simulation result shows that the mud rammed will have warmer than the concrete building. Now on the energy demand and actual consumption, so from here what we can see is actual consumption is lower than energy required. So actual consumption is calculated based on the monthly electricity bill. So that the gap between these two, what we can understand is that maybe in order they must be using a localized heating system like Bukhari, or as usual they might be using, or we might be using the warmer clothes or thicker clothes while even staying inside the building. And then what we can understand from here is that energy demand in RAM wall is lower than energy demand in the concrete buildings. So the final finding is that the total mud RAM building will save annual heating energy by 27.41%. On the discussions, so the first one is interpretation of results. So what it says is saving energy potential of mud RAM wall confirms the indigenous design can outperform the modern methods in energy efficiency and comfort for the occupants. And then with regards to the practical implications, so encourages policy measures for promoting the traditional materials in the new construction and retrofit the national energy. So reinforces, so sorry, the last one, Empirical validation provides evidence supporting the modern wall over the concrete wall. And then simulation insights. So we get the valuable data from the simulations. And last, the policy relevance. So some emphasizes the importance of the integrating findings into the energy policies for sustainable architectural practices. So the future research directions are further studies should explore hybrid wall system, long-term durability, moisture performance, and scalability across different climatic zones. So these are the references, and thank you very much for providing this platform. Thank you.
Thank you, Samdan, for this very interesting and relevant research, where the key takeaway for me personally was how we have a lot to learn from our traditional and indigenous way of life. Thank you to all our presenters. We will now open the floor for our Q&A session. May I invite our keynote speaker, Professor Yong, to moderate the session, along with the presenters of session 1, Sawang Nidup, Dr. Mamta Chetri, Samdan Lendup, and Dr. Leela, who's online.
Yep. Can you? All right. Thank you to all our distinguished presenters for a fascinating and diverse set of insight. So we have actually heard perspective across material science, biosecurity, genomics, and even sustainable building design. So all align with our theme of integrating STEMs for societal impact. So I might now open the floor for questions. Please raise your hand, introduce yourself briefly, and specify which speakers your questions is directed to. Anyone? Oh, fabulous. Please. Uh, the, the mic, please, in front. Thank you.
Hello, everyone. Thank you, everyone on the panel, for your presentations. I'm Annie Gowing, and I'm from the University of Melbourne. And I actually have a question for the wonderful speaker on the right, who finished with that very interesting and provocative quote. And I'd just like you to unpack it for us, because I'm very interested in what the meanings that sit under that. Thank you.
Please. Yes, thank you very much for your question. I was kind of expecting that even during my presentation, and I think that's why I said maybe it's a bit controversial as well. But I think the rationale and the idea behind this is that the worst thing that we can do with timber is to burn it. And the next is to export it, I think. And then maybe a better way to utilize this is to make furnitures. But once people feel the value of that material, I think the idea is that then they begin to protect and preserve it as well. So I think that's why I said also in my presentation, the building sector provides the best avenue and scope for the high value utilization of this material. So I think there are also research and studies now kind of suggesting that in places where timber buildings are being promoted in parallel with sustainable forest management practices is a stark increase in the net forest coverage as well, I think. So I don't know if I Yep. Yeah.
Thank you. Yeah, please.
Thank you very much for those very wonderful presentations. It's a pity that Dr. Mamta couldn't complete her presentation. So I'd like to request her to share the relevance for Bhutan, the research she's done.
Please.
At the University of Sydney, we had this serial rust surveillance program for a century, and it has worked well, and it is a world model for serial rust surveillance program. And for this, as we are opening door in GMC, I would like to suggest that we also integrate in Bhutan, like with RAB, and biodiversity, because we have biodiversity hotspot here, and when we open the door to outside world, there was that pathogen coming in as well, which will also impact our biodiversity. I would like integration happening between RUB with research happening, just like Sydney Uni, and integrate it with Ministry of Agriculture, including biodiversity, organic farming, and NPPC. All this integration, if they come together, just like we did in University of Sydney, we have protected cereal industry for a century. It will happen in Bhutan as well. That was my recommendation for Bhutan.
Thank you. Okay.
Hello, sir.
Yeah, please.
My question is addressed. I want to put forward this question to Mr. Something. So, is there a way or a potential that the ramp construction can enhance Bhutan's climate resilience by reducing the reliance on the imported construction materials and fossil fuel-based energy? Thank you.
Sorry, I could not hear your question, please.
So, my question is that is there any way or potential that the ram earth construction can enhance Bhutan's climate resilience?
I may not be like really give you the specific answers, but to some extent, yes, since the using of the local material available, especially like mud-rammed walls, will like save the energy, especially the energy, and therefore, like we can like it can also help in the climate change as well. So, but again, on other hand, in long term, if you talk about the durability, then I think again that's an issue when we compare with the concrete walls. Thank you.
Thank you. Yep, please. Yeah. The mic is not working for the gentleman.
Hello.
Yep. Thank you. Please.
My name is. Hello. My name is Bim Rai. I'm from Australian National University. My question is to Dr. Mamta. Your study is mainly based in Sydney, and I presume the humidity, altitude is obviously different for Sydney and Bhutan. So will that affect the approach if we apply your knowledge in Bhutan?
Thank you, Dr. Dan, for the question. Yes, it is different, but the pathogen, they survive in different environment, and in Sydney, the pathogen, in Australia, the pathogen are different, but in Bhutan, there are pathogen, and in Bhutan, there is a threat. If you look at the rust, there is sexual recombination happening because we have Berberis in Berberis around. There, like sexual combination happens in Berberis because it has got two hosts. If I talk about rust disease, there is two hosts. Sexual recombination happens in the secondary host, and then after that it goes into the plant material. And why I'm stressing on sexual reproduction is that there will be new variants which can overcome the resistant genes in variety which we grow in our area. So it will be like, I mean, even if the climatic conditions are different, but we have different of threat in different climatic condition also. I think that model should work for Bhutan as well.
Thank you. Oh, please,
Thank you very much, Professor Yong. My name is David Wong from Australia, Notre Dame University. I have a question for our colleague here on the right-hand side regarding the timber species. I'm sorry, I forgot your name. You have presented some data there on the characteristics, stiffness, and strength of the timber species that you're looking at. I believe yours British spruce. I wonder how this timber and maybe other species can be adapted to the Bhutanese weather. And I ask this question because I was quite honored to stay in a very nice hotel near to Paro upon arriving near Taksang. And it was completely made of timber. But then it was also, a little bit warped here and there. So I'm wondering regarding the moisture content, retention, maybe even the propensity to swelling and fungal decay, maybe even the point of catastrophic failure in a fire, and so forth. So are these other characteristics also favorable for the Bhutanese conditions? Thank you.
Yes, so in as much as being an advocate for timber, I think we'll also have to accept its limitations. And yes, I think no material is perfect. Concrete has its own flaws, steel has its own flaws, but the idea is to how to address and mitigate these challenges, I think. I think some species are naturally well adapted to certain climatic conditions. And I think there's a lot of work that we need to do to identify what species will be best suited for what application purposes in what regions. And as maybe if I've not been clear in the beginning, I think this work has just started. I think elsewhere in Europe and even in Australia, I'm sure they are already 30, 40 years down the line. I think we are just at the beginning stage of this exploration. But yes, definitely. But one point I would like to make in terms of fire, I think it's quite unique. And one big advantage with timber in terms of its fire performance is that it's the misconception that because we are quite used to seeing timber being burned for fuel and for heat. I think the general conception is timber is quite combustible. Yes, it is. But the benefit also is that it's also, because as it burns, we would have seen there's a black layer of char being formed, and that char acts as a natural insulator. So in the design, what we would do is, if, for example, this column needs to be like 2 inches square, a good practice would be to provide a sacrificial layer of an additional 1 inch to act as a fuse, so that when there's a fire, that 1 inch burns and a char layer is formed, and the main core that needs to be there to support the load still stands. So yeah, I mean, it's a big topic. I think maybe we can discuss further on this. But yes, definitely there's a lot more to be done. And I think we're just making the initial inroads into this. I hope I've satisfied your queries.
Thank you. Thank you for. Please.
Good morning. My name is Kezam Sumo. I'm from Department of Agriculture. My question is for Dr. Mamta and Dr. Riza. I would like to ask if, since I think, unfortunately, Dr. Mamta couldn't finish her presentation, but it's a very interesting area for the department. For us, I think what we are looking at is, of course, the control of rust. You need to use a lot of fungicide. And if we could have a good surveillance system, that would help us to reduce the use of fungicide in the crop. Have you looked at any biofungicides that could be used in control of rust? And for Dr. Rizal, I wanted to ask, I think you mentioned about your biocontrols possibility, and I wanted to ask if there is any possibility for the Bhutanese researchers to collaborate with your universities in research in plant pathology, especially in biological matters, materials that could be used for pest control, fungus, as well as for herbicides.
Shall we ask Dr. Rizal to take the question? Dr. Rizal, would you mind to take the questions? Can you hear us?
Thank you very much. Can you hear me?
Yes, perfect.
This is an area I'm working currently. Of course, not directly related to rust, but at the moment I'm working on growing plant pathogen on growing crops in Australia. And what I am doing is, in order to control the Fusarium for example, Fusarium pseudograminarium cause crown rot in wheat. So what I'm doing is I am isolating all the microbes, both including the bacteria as well as fungi, from that system, and then screening them individually to see if there is any antimicrobial activity against that pathogen. And after we screen the isolates, we do a secondary metabolite testing, and then that's a pipeline to produce some secondary metabolites to control that pathogen. So that is under pipeline, and I'm actively working in this area. And sometimes, because biocontrol has been really a hot topic at the moment, even in Australia. So I think there is possibility, but I would suggest having your own isolate. Like we cannot guarantee the Australian isolate or antimicrobial fungal or bacteria would be really effective in Bhutan's climate. So it would be a really good research to carry out in Bhutan, like looking into that system, getting the microbes from that system, and then trying to control the pathogen in that system. So that's my recommendation. Thank you. Hope I clarified your doubt.
Thank you, Dr. Rizal. I'll pass the floor to Dr. Munta.
Regarding the collaboration, we have been talking with Ministry of Agriculture. I had a Zoom meeting with Director of MOE, and being in agriculture myself for about 10 years in Bhutan, I know the constraint, and I had mentioned to Department of Agriculture that, their main concern was that there is rust disease, and they don't have material which can resist this variants which are going on in their field. So we have made arrangements saying that whatever we have developed in Australia, we can bring back to Bhutan and then do a collaborative research. But we are looking for funds right now so that we can integrate, what we have done in Australia. We usually in Plant Breeding Institute, we develop varieties which can resist the upcoming variants. So we also have this pyramiding of genes going on. So we have those material and we are talking right now to collaborate.
Thank you. Thank you. Thank you, Dr. Mamta. Oh, please, the gentleman.
Good example, everyone. I'm Sang Chu, a final year architecture student from College of Science and Technology, and my question pertains to Mr. Santan. Firstly, thank you for the insightful presentation and the very well comparative analysis on thermal performance. My question is like, in your study, you focus on thermal performance using climatic data. But in real life, is climatic data alone sufficient to make a material selection, or building as a multidisciplinary system, should we consider other aspects like aesthetics, functionality, and structural performance as well? Thank you.
Please pass me the mic.
Thank you. No, but I'm not really sure what you're supposed to get from me. But as far as I'm concerned, first of all, like if you have to do simulation with buildings, the important the data required is climatic data, which is very important. And then as of now, the greatest challenge that we are facing is not getting the climate data. And then other thing, of course, if you just go with the own architecture detail and all, like very important thing in order to see, I mean, the content, the energy, any sort of energy inside the building is, I think we should really take care of the thermal bridges in order to fit whatever the architecture details that are required in the building. And then in terms of material selection, yes, true. If we have all this, the forecasting of the weather data, based on that, if we can really find the solutions for that, because I also have a small experience while collecting the data on the insulation for the building materials, like especially with the cement and then the sodas. So while collecting all those data, it was quite challenging for us to really get the appropriate data as well as the climatic data for studying further to say that this material is suited for this climatic condition. So thank you very much.
Thank you. I think that the time is actually run out. I think that it will be good for us to end the sessions here. But again, thank you all for your thoughtful questions, and to our presenters, for their excellent responses. So these sessions, has shown that how STEM, when integrated across disciplines, can lead to meaningful societal impact. So we now break for tea. Thank you. Thank you, everyone.
Rasla, thank you for this enriching discussion. This concludes the session on Thematic Area 2. We will now take a 20-minute tea break. Please return on time at 11 a.m. as we continue with Thematic Area 4.